welcome back to my channel, Explaining Biology Clearly. Today I'm going to talk about prokaryotic cells, a little bit about their structure, some of the things that you find inside the cell. Now, I have had a look in a previous video at animal and plant cells and their structures and some of the similarities between them and some of the differences. They are both examples of eukaryotic cells, cells with a true nucleus. So have a look at that post if you haven't already. I will be having a look at some of these organelles in future videos in more detail, maybe a different video on each of these organelles. Please feel free to ask any questions or suggest any other video titles. But today we're going to have a look at what these prokaryotic cells are and what they look like. So the word pro refers to before, and the word karyon or karyotic refers to nucleus. So these are cells that are thought to have evolved or arisen before the evolution of a nucleus. So these were the first life forms on Earth. They are the bacteria, so the only cells that have, the only living things that have these cells are bacteria. And the ancient bacteria, the archaebacteria. So bacteria have prokaryotic cells. They're the only type of prokaryotic. They're the only living prokaryotic cell on Earth. Whereas if you compare that to the eukaryotes, you've got animal, plants, fungi and Protoctista. They're all eukaryotic. So there's more than one kingdom of eukaryotics, but there is only one kingdom of living prokaryotics. So having a look at their structure then, so if we draw a bacteria here, around the outside of their cell you find a cell wall, and this is made of something called peptidoglycan or murine. I tend to call it peptidoglycan, but you might refer to it as murine. And that's obviously you find a cell wall in plant cells and fungal cells and some Protoctista, but it's not made of peptidoglycan. It's made of cellulose in plants and chitin in fungi. So the substance that makes it is different. And then underneath the cell wall you have your cell membrane. So that's that lovely purple structure there, the cell membrane. Same structure, same components, made of the same substance as in all the other cells. This funny infolding that I've drawn here, that's something called a mesosome, and it increases the surface area of the cell membrane. It's folded into the cytoplasm. There's the cytoplasm, by the way, just there. So the cell membrane is folded into the cytoplasm, and it has enzymes on its surface which carry out reactions like respiration or photosynthesis. So because these cells don't have any membrane-bound organelles, they don't have mitochondria or chloroplasts or Golgi bodies or endoplasmic reticulum or nuclei, vesicles, vacuoles. They have none of those, no membrane-bound organelles. They can't carry out the chemical reactions that take place inside those organelles. But prokaryotic cells still need to respire. They still need to break down glucose. Some bacteria still photosynthesize to produce their own sugars. So they need these enzymes in order to carry out those reactions, the reaction of respiration and photosynthesis, for example. So no membrane-bound organelles, that's really, really important thing to remember about these. So we've got no nucleus here. So instead of a nucleus, you're going to still find DNA, but it's just free in the cytoplasm. And that area that it's found in is sometimes referred to as the nucleoid, the area that you find the DNA. And it's one big loop, circular DNA. Just write that down. It's one big loop of circular DNA, not like in the eukaryotes where it's linear. It's a straight line. It's like a chromosome, this typical X-shaped chromosome. It's in a line. This is circular DNA. As well as that main loop of DNA there, the bacterial cell will probably have smaller loops of DNA called plasmids. So these are small loops of DNA scattered in the cytoplasm there as well. And because the cell still needs to make its own proteins, still needs to make its own enzymes, etc., it does have ribosomes in order to assemble those proteins. So you might be wondering why I've said there's no organelle, no membrane-bound organelles. Well, ribosomes aren't membrane-bound. They don't have a membrane around them. They're just organelles. They're just structures that carry out a specific function, but they don't have a membrane around them. They're just made of ribosomal RNA, no membrane around it. So that's the basic structure. You may find in some of these cells, you may find an extra layer around the outside called the capsule or slime layer. Sort of a mucousy layer around the outside. It might help it adhere to surfaces or to other cells. So that would go all the way around the outside of the cell. And in some cells you might find one or more flagella. So I've drawn just one there, so it's a flagellum singular, flagella plural, and that can beat and help to move the prokaryotic cell in a liquid medium. So that's the basic structure. I will post some videos on each of these things separately, but please feel free to ask any questions, and I hope that has helped you to see the difference between the eukaryotic and the prokaryotic cells. So thank you very much for listening to my video. Please like and subscribe and have a look at some more. Thank you.